Alright, hi everyone, my name is Jasmine, I'm a junior, and today I'll be presenting on the unsung heroes of the brain, bioavailable phenolic acids and metabolites that guard against oxidative stress. So first, what is oxidative stress? Oxidative stress is the imbalance between molecules called ROS and antioxidants. ROS is the acronym for reactive oxygen species. These molecules are oxygen-containing compounds that are byproducts of cellular metabolism. They're extremely reactive molecules because they have unpaired electrons in their outer shells, which leads them to become chemically unstable, and so they're eager to steal electrons away from other molecules. A subcategory of ROS are free radicals, and these are the ROS molecules explored in this paper. Free radicals specifically are an atom or molecule with an unpaired electron. In a healthy cell, ROS is used for redox activity, meaning ROS can be used to oxidize or reduce molecules for cell signaling purposes. This change is often minor and reversible, so it doesn't cause permanent damage. Next, we have antioxidants. Antioxidants are molecules that can neutralize unstable free radicals through contributing an electron or hydrogen atom. And in a healthy cell, there's a delicate balance between ROS and antioxidants. But under oxidative stress, ROS levels are so elevated that antioxidants can't control them anymore, thus leading ROS to cause irreversible damage to key molecules like DNA or proteins. This disrupts cell function and leads to cell death or apoptosis. And neurons, specifically, are very susceptible to oxidative stress because of their heavy reliance on oxygen. Thus, oxidative stress is a hallmark of many neurodegenerative diseases, such as Alzheimer's and Parkinson's. But our body does have natural ways of guarding against oxidative stress via polyphenols. Polyphenols are molecules that have antioxidant properties, and they're often found in beverages, fruits, and vegetables. They're very common in red wine, pomegranates, grape, and cocoa. These molecules help protect our cells from damage, inflammation, reduce oxidative stress, and improve overall bodily function. But they do have some major weaknesses, mainly because of their size. Since they're fairly big molecules, they have a very poor bioavailability, meaning they can't cross into the body's circulation, aka the blood, that well, and can't reach all parts of the body that well either. This is especially prevalent in the brain because the brain's blood vessels have a thicker inner lining called the blood-brain barrier. This barrier acts as a filter for molecules and substances entering and exiting the brain, and only molecules that are small enough or lipid-soluble can pass this barrier, which polyphenols cannot. But they do have natural ways of overcoming this disadvantage. Since polyphenols are taken into the body through food, they can also be digested and broken down by gut bacteria. These smaller molecules, called metabolites, are the ones that can enter circulation and reach different parts of the body. But because of the sheer number of metabolites in the blood, it's really unclear which ones are the ones providing antioxidant effects. It's even more so unclear in the brain because of the blood-brain barrier. In fact, only low concentrations of metabolites have been detected in the brain. And then these few metabolites are gallic acid and ellagic acid. The fact that they can cross the blood-brain barrier suggests a potential role in neuroprotection, but it doesn't actually confirm that they are the ones doing it. So now we see a knowledge gap that this paper aims to address. This paper aims to use a panel of known polyphenol-derived metabolites that are bioavailable. So if we do see neuroprotection, then we can say for sure that these metabolites are the ones mitigating oxidative stress. Next, previous demonstrations of metabolites' neuroprotective effects were used in biologically irrelevant conditions. So this study aims to use relevant conditions of metabolites that you would see in an organism to therefore provide more direct knowledge about the neuroprotective role. And so to do so, this paper cultured SHSY5Y cells. These cells come from a neuroblastoma cell line. Neuroblastoma is the cancer of immature neurons. And they use the neuroblastoma cell line because they can then mimic neurodegenerative diseases like Alzheimer's. To introduce oxidative stress, these cells were incubated with hydrogen peroxide. Hydrogen peroxide can induce oxidative stress because it can react with iron ions and then generate the highly reactive OH radical that has electrons in its outer shell. This is the mechanism for oxidative damage. To collect data, cells were pre-treated or co-treated. Pre-treatment included incubating cells with each test molecule for 6 hours, and then a 24-hour incubation with hydrogen peroxide to reduce oxidative stress. And then with co-treatment, both hydrogen peroxide and the test molecules were incubated together. So first, let's get into figure 1. In figure 1, we're looking at the structure of the 19 polyphenol-derived metabolites tested in this study. What's important to note here is that each metabolite contains a phenyl group. A phenyl group is a carbon-hydrogen ring with a hydroxyl group attached to the end. This allows for antioxidant properties because phenyl groups can donate hydrogen atoms or electrons to neutralize free radicals. On the top, we can see 4 emphasized structures. These 4 were found to have the greatest neuroprotective effects, so scientists decided to emphasize them. Then lower, we can see the rest of the metabolites, and these are less significant. We can see that there are a lot of groups derived from the same polyphenols. For example, this one group, that's one group, and then that's one group. And so a lot of them have really similar structures. For example, the only difference between Euro B and Euro A is that Euro B has a hydrogen, while Euro A has a hydroxyl group from the R1 side chain. Next, if we get into figure 2. Figure 2 measures cellular viability via an MTT assay. So on the y-axis, we're looking at cell viability measured in %, and on the x-axis, we're looking at the different treatment of cells. All these graphs have a healthy control. These cells do not have any metabolites added and also no oxidative stress. And then we also have a stress cell control, which has only been incubated with hydrogen peroxide. And we can see that all of the stress cells have major cell viability decreases of around 40 to 50%. These figures also differentiate between pro-treatment and co-treatment as well. So now in figure 2a. Figure 2a, these first 5 metabolites, we can see that there are no significant increases in both pre-treatment and co-treatment. But with these last 4, DHPPA, DHPAA, and gallic and ellagic acid, there are significant increases in cellular viability in both pre-treatment and co-treatment. In figure 2b, we're testing the other metabolites used in this study. What's important to note here are that the pre-treatments see very significant increases in cellular viability, except for Euro B glucuronide over here. However, none of the co-treatments showcase a significant increase. So then in figure 2c, scientists chose to further evaluate the four best performing metabolites. But this time, they're being evaluated at lower biologically irrelevant conditions at 1 and 5 micromolars. We can see at 1 micromolar, there isn't really any significant increase. But at 5 micromolars, we can see significant increases of all metabolites except for ellagic acid. So now that scientists know what metabolites had neuroprotective effects, they wanted to evaluate why or how. And so the first step in doing so was testing the amount of intracellular ROS in the cell. If you recall, in oxidative stress, we should see elevated ROS levels. And so we can see that the stress cell control majorly increased the amount of ROS. And then the metabolites significantly decreased the amount of ROS with both pre and co-treatment, except for Euro A and co-treatment. Next, scientists evaluated redox levels. In oxidative stress, ROS causes damage to the cell instead of participating in its usual activity of redox signaling. Thus, we should see a decrease in redox activity in stress cells, which we do. And then we can see the metabolites restored this amount, except for Euro A in both pre and co-treatment. And before we get into later figures, it's important to understand a little bit about apoptosis. So what is apoptosis? It's programmed cell death. Cells that are marked for apoptosis are often mutated or damaged beyond repair. And since oxidative stress causes intracellular damage and usually can't be repaired that well, oxidatively stressed cells often go through apoptosis. Now, the actual cascade of apoptosis is fairly complex, but the only parts that we really need to understand here are caspases. Caspases are a family of enzymes that act as basically cellular scissors to cleave specific proteins and control apoptosis. First, let's take a look at caspase 3. Caspase 3 is often considered the crucial executioner of apoptosis because it does most of the cleaving. And we can see that it's activated from both the extrinsic and intrinsic pathways, so if caspase 3 is activated, that's a major sign that apoptosis is occurring. Speaking of the initiators, let's take a look at caspase 8. Caspase 8 is often considered a crucial initiator because it can activate caspase 3, the crucial executioner, from the extrinsic pathway. Extrinsic pathway just means that this pathway starts on the extracellular surface of the cell. Similarly, caspase 9 is also an initiator, but caspase 9 initiates caspase 3 via the intrinsic pathway, which means this pathway starts from intracellular regions. Apoptosis also has a major differentiation between early and late stage apoptosis. Early stage, the cell is generally still intact, maybe still functioning, maybe it's just been marked for apoptosis and caspases are just being activated. For late stage apoptosis, this is when the cell is non-functioning, dead, and needs to be cleaned up. Think DNA is fragmented and the membrane is breaking down. So next, in figure 3, on the y-axis, we're measuring the percent of apoptotic cells. And so from both these graphs, we can conclude total apoptosis levels and also a difference between early and late apoptosis. And then again, on the x-axis, we're looking at the different treatments. Similar to the other graphs, healthy cell control, and then also a only stressed control. So if we look at figure 3a, it's the pretreatment of the cells. We can see that hydrogen peroxide increases apoptosis by around 20%, with massive increases in late apoptosis. This means a lot of cells are non-functioning and need to be cleaned up. But we can see the metabolites decrease this total amount of apoptosis, especially late apoptosis, meaning they are successfully mitigating oxidative stress and leading to less cell death. And then figure 3b looks at co-treatment. Again, we can see that these four metabolites, DHPPA, DHPAA, gallic, and ellagic acid, are reducing amounts of total apoptosis. However, we can't say the same for UroA as the error bars overlap, signifying that these results aren't significant enough. And then, figure 4 looks at caspase activation. On the y-axis, we're looking at the percent of cells or caspases that are activated. And then on the x-axis, we're looking at the treatment of different metabolites. Again, healthy cell control and a stress cell control. In figure 4a, we're looking at the pretreated cells. So we can see that the stress cells show a significant increase in caspase 3 and caspase 9. This makes sense because oxidative stress causes intracellular damage, so the initiator caspase from the intrinsic pathway, caspase 9, is being activated more. And we can see the metabolites cause significant decreases of activated caspase 3 and 9. But if we take a look at 4b, 4b is looking at co-treatment. Again, stress cells show elevated concentrations of activated caspase 3 and caspase 9. And these last four show major decreases in these activations. But again, we can't say the same for UroA as error bars overlap. And so, for some conclusions and significance, this paper addressed the knowledge gap. It was unclear which metabolites are responsible for neuroprotective effects against oxidative stress, especially in the brain because of the blood-brain barrier. And lots of studies before this paper used concentrations of metabolites that aren't biologically relevant. They were either too high or too low. So what did this paper find? This paper found that 4 metabolites, DHPAA, DHPPA, gallic, and ellagic acid, were found to have the most potent effects against oxidative stress among the 19 metabolites tested in this study. What was specific about these 4 metabolites were that both pretreatment and co-treatment were found effective against oxidative stress. We also found that UroA, this metabolite, was also effective, however, only pretreatment. At co-treatment, didn't produce significant enough results. And so what does this paper tell us about our diet? Well, if you eat a lot of foods that contain polyphenol precursors to these effective metabolites, then that could have a contributing factor to mitigating oxidative stress and the onset of neurodegenerative diseases. These foods include cocoa, strawberries, tea, and pomegranates. For some further directions, this paper only tested results of mechanisms, like what's being decreased or increased, but not the main mechanism itself. So that could be a road for further exploration. For acknowledgements, I would like to thank the Journal Club staff, especially the team that worked with me closely on this paper, Mr. Maxwell, and all of the teachers giving extra credit. Thank you. Do we have any questions?
Do you think like the cancerous nature of the cell line might have affected your results and made them different than if you had like tested on like cells of different neurodegenerative disorders?
Well, the main reason that they used a neuroblastoma cell line is because if they were to use cell lines from like different neurodegenerative diseases, first of all, those aren't really like widely available.
I'm just saying that it like affected your results. Do you think it had any effect?
I wouldn't think so, because a neuroblastoma cell line is very similar to many of the neurodegenerative disease cell lines, so I think they would produce around the same results. I think it was mainly just like a problem of getting those cells in the first place. Yeah. Brian?
How do metabolites differ in their ability to inhibit the caspase activation?
So we saw major significant decreases from 4 metabolites. So DHPPA, DHPAA, and then gallic and ellagic acid had really significant decreases. The paper doesn't really get into mechanisms that much, which is why it's probably a lane for further exploration. But the main thing, just to see, like with caspase, like activation and deactivation, is just like these 4 work really well in pretreatment and both pretreatment and co-treatment, while UroA only was able to decrease really significantly in only pretreatment.
Yeah.
Okay. Yeah.
Did they look at any of these in combination?
No, only by themselves, yeah. Okay, thank you.